From this chapter, we're going to learn metabolism. The first metabolic pathway is glycolysis. Glycolysis is a pathway to break down glucose and generate ATP and electrons. In eukaryotic cells, glycolysis takes place in the cytoplasm. Glucose is a major fuel for our body. In fact, our brain and red blood cells rely solely on glucose as a fuel. The pathway to synthesize glucose is called gluconeogenesis, that we're going to talk about in the next chapter. Chapter 16 contains 5 sections. Section 1, glycolysis is an energy conversion pathway. Section 2, NAD+ is regenerated. Section 3, fructose and galactose enter the glycolytic pathway different than glucose. Section 4, the glycolytic pathway is regulated. Section 5 is a case study. At the beginning of the semester, we watched the video. I made this screenshot and call it this big picture about the metabolism and said that each arrow on this big picture could be a chapter. Let's see, where is the chapter 16 is on this big picture. You see, it's this from glucose to pyruvate. This is another big picture. The glycolysis, it turns a 6-carbon glucose break down to 3-carbon pyruvate. 1 glucose break down to 2 pyruvates. This process generates ATPs and also electrons. The yellow lines indicate electrons flow. The electrons from glycolysis flows to other pathways in cellular respiration. The glycose happens in cytoplasm. This is a simplified scheme, helps us to understand glycolysis. It starts with 1 glucose compound inside the cell cytoplasm. The glucose goes through a phosphorylation, and the phosphate is added to the glucose. It causes ATP to turn to ADP. The yellow ball represents 1 phosphate. The second phosphorylation adds another phosphate to the 6th carbon, consume another ATP. This whole process consumes 2 ATPs, add 2 phosphate function group onto this 6-carbon compound. It's very unstable. It breaks down to 2 identical 3-carbon compound. This 3-carbon compound has very high energy. It goes through a spontaneous reaction, oxidation reaction to become a less energy compound, and this process release 2 electrons. Each C3 release 2 electrons, and NAD+ as an electron acceptor change to NADH. So this process produces 2 NADH. The 2 identical C3 with each has 2 phosphate function group release phosphate and turn ADP to ATP. That's how ATP is produced. This process total produce 4 ATPs. It ends with 2 pyruvate molecules. So the whole glycolysis is 1,6-carbon glucose turns to 2,3-carbon pyruvate. This whole process consumed 2 ATPs but produced 4 ATPs. So total production of ATP is 2. And also produce 2 NADH products. This is a figure in the textbook. I introduced the previous slides so that you have a big picture of the pathway and won't be overwhelmed with the details. I printed this figure as a handout. It's recommended that you print it yourself and use it as a reference in the future. As I mentioned before, the glycolysis takes place in cytoplasm. It's a process converts 1 molecule of glucose to 2 molecules of pyruvate with generation of net 2 molecule ATP and 2 NADH. It can categorize into 2 stages. Stage 1 consumed 2 ATPs to trap the glucose in the cell and modifies it so that it can be cleaved into a pair of phosphorylated 3-carbon compounds. Stage 2 oxidizes the 3-carbon compound to pyruvate while generating 2 molecules of ATP. Note 1 glucose converts to 2 pyruvate, and 1 pyruvate produces 2 ATP. So 1 glucose should produce 4 ATPs. And on this chart, on the stage 2, it's all about 1 3-carbon compound, and you have to calculate by times 2. Step 1. You can see the glucose is converted to glucose 6-phosphate, and that need a phosphate. It comes from ATP. So ATP is consumed in this step. And the enzyme is hexokinase. This step is irreversible. You can see from the arrow, it's just one arrow, one direction, and irreversible. Step 2, see the arrow? There's 2 arrows, so it's reversible. Step 3, it adds another phosphate onto this compound, so it consumes another ATP. Now this compound has 2 phosphate. You can see the structure fructose 1,6-biphosphate. This compound has a high energy, unstable. It will break up to 2 3-carbon compounds. This process from step 1 to 3 use 2 ATPs. And up to step 4, the glucose is trapped inside the cell. It cannot go back to glucose anymore. Step 5, it cleaves into 2 interconvertible 3-carbon molecules. So fructose 1,6-biphosphate is cleaved into 2 interconvertible 3-carbon molecules. In this glyceraldehyde 3-phosphate, it converted into 3,1,3-biphosphoglycerate. And this step is the oxidation step. How do you know it's the oxidation step? Normally, when oxidation happens, it will involve a NAD plus as an electron acceptor, a free phosphate, and a NADH. The NADH carries a pair of electrons by combining with hydrate. Number 7 is reversible, and number 8, 9 are all reversible, except number 10 is irreversible. You see the arrow is single arrow, and ADP is converted to ATP through pyruvate kinase, and that eventually produced the pyruvate, and that's the end of this glycolysis pathway. So in general, this pathway, it convert 1 glucose into 2 pyruvate. From this, look at the stage 1, there's a few irreversible steps. In the stage 2, there are only 1 irreversible step. That's the step 10, when the phosphoroenopyruvate turn to pyruvate. It generates 4 ATP. Stage 2 indicated it generates 2 ATP, but remember, the glucose break up into 2 3-carbon molecule, so there's 2 pyruvate is produced, 4 ATP is produced. See, that's times 2, you have to do the calculation, and therefore the net at ATP is 2. Here's a summary of the glycolysis. It totally 10 steps. For stage 1, the first 5 steps, glucose plus 2 ATP consume 2 ATP and produce 2 GADP. In stage 2, the last 5 steps, 2 GADP produce 2 pyruvate plus 4 ATP, and that is 2 ATP. This table lists in detail of the 10 steps in glycolysis. It's a very good reference for each reactant, product, as well as enzyme in each step. I highlight the 3 kinase enzymes. These 3 enzymes play vital role in glycolysis regulation. We're going to talk about them later. The textbook explains every reaction in glycolysis pathway. I think after watching the video, look at the above table, we have a pretty good idea on all the reactions happen. Here I want to point out one key point about all the reactions. You see in the table, there are some reactions are irreversible, some are reversible. Everything happens here is about energy, like we learned in the past. When the delta G is negative, the reaction will happen spontaneously, which are irreversible reaction. And if some reaction need energy, then it's reversible. This figure I used multiple times. I want to use it again. It's about energy. It's about how you determine if a reaction is producing ATP or consuming ATP. In this figure, the ATP is in the center. Compounds above ATP has higher energy. It intends to give a phosphate to ADP turn to ATP. So higher energy compound intend to produce ATP. Lower compound, the one below ATP, the glucose 6-phosphate, glycolysis 3-phosphate, they have lower energy. If they want to move up in energy, they need get a phosphate from ATP. They need to consume ATP. So anything has energy lower than ATP would need to consume ATP, and the compound above an ATP in this figure will produce ATP. So energy, everything is about energy. For this one, I just want you to have an idea about energy, free energy. You can see here some reaction, like the first step, the reaction is that delta G is minus. So the first reaction is irreversible. And if the delta G is positive or negative in a small amount, that means it could be either way is isomerizations. And then if it's positive, it just need to consume an ATP. And if it's negative, then it's spontaneous, irreversible. So energy, everything is energy. This is the total equation for glycolysis. The net reaction for glycolysis is 1 glucose plus 2 free phosphate, 2 ADP, 2 NAD plus, turn to 2 pyruvate, 2 ATP, and 2 NADH, plus 2 proton and 2 water molecule. The conversion of glucose into pyruvate generates ATP, but for ATP synthesis to continue, NADH must be reoxidized to NAD+. This vital enzyme is derived from the vitamin B3. NAD+ can be regenerated by further oxidation of pyruvate to carbon dioxide or by the formation of ethanol or lactate from pyruvate through fermentation. In order to regenerate NAD+, pyruvate, a 3-carbon compound, has to be further metabolized. Electrons formed from NADH are taken away to form NAD+. In order to make the glycolysis to continue, NAD+ has to be constantly regenerated. The metabolism of pyruvate can be a anaerobic fermentation, like the winemaking to produce alcohol, ethanol, release carbon dioxide, or like the milk fermentation from pyruvate to form lactate. When oxygen is present, it will undergo the citric acid pathway, and the electron transport chain. We're going to learn that in section 8 and 9. In these circumstances, oxygen accepts electrons and protons to form water.